Okay, so what we're going to learn in these topics, homeostasis in plants, we're going to look at how these stomata, they will have the daily rhythms, rhythms of opening and closing, and also respond to the change in environmental conditions to allow diffusions of carbon dioxide, as well as to regulate the water loss by transpiration. So it means that these stomata, they are not permanently open or permanently closed. Okay, so they actually, we can have the homeostasis, or plant can have this homeostasis to change. Okay, these are the diameter or the pore diameter of the stomata. Okay, and in this case, we do know that stomata is the pore. So the one that control the pore size is this a pair of cells. So this a pair of cells is called as a gut cells. Okay, so we're going to look at the structure together with the functions of the gut cells, and we're going to explain the mechanism by which how this gut cell can control the opening and the closings of the stomata. Okay, so when the plants actually undergo the water stress, and when we use the word water stress, means that the supplies of the water, okay, cannot meet the demands of the plants. So the plant will undergo this water stress. Okay, most of the time it's because of the transpiration. Too high the rate of transpiration, they lose more water contents as a water vapor compared to what they can get from the soils. Okay, so in this case, then we have to look at one of the hormones called abscisic acids, or in short, we call it as ABA. So this hormone actually can cause the closure of the stomata, therefore reduce the rate of transpirations. Then the plant will actually manage to survive, okay, at this condition, even though they may not be able to mix the means to have the supplies of the water, because they can reduce the rate of transpirations. Clear? So we look at this opening and closing of the stomata. So first of all, we do know that plants have to maintain the constant internal environment similar to the animal. So in this case, in order for them to carry out photosynthesis, they need a constant supply of the carbon dioxide. So linked with the photosynthesis, we do know this. Okay, and who control the entry of the carbon dioxide into the leaf? Basically, is the stomata. Okay, stomata is the one. So what is the stomata? So stomata, they are the pore between the gut cell. Okay, but normally we use the words, when we talk about stomata, we refer to the two gut cells and the pore between them. But in fact, stomata basically is the pore between the gut cells. And the gut cells, they are highly specialized cells. So it means that in this case, you can see that they actually regulate the pore size. And why is it highly specialized? Because they respond to the wide range of environmental stimuli, and based on this environmental stimuli, then they control the pore size, or this the diameter of the stomata, so that therefore they can regulate the internal atmosphere of the leaf. So whether we need to have more CO2 enter, or we need to stop the CO2 enter, and this can be regulated by the gut cells. So since the gut cell is so specialized, then we look at how the gut cell look like. Okay, so for every pore, if you look at this, for every single pore is controlled or is regulated by a pair of the gut cell. So if we zoom into this gut cells, then we can see that they have this uneven thickness of the cell wall. If you look at it, so thinner. Okay, can you see that? Thinner at the wall that away from the pore, thicker. Okay, wall that's the ones that actually form the pore. Okay, so highlight gut cells, they have uneven thickness. Okay, in terms of the cell wall, the adjacent to the pore or near to the pore, they are very thick, whereas the wall further from the pore is thin. And they have the bundle of cellulose microfiber arranged as hoops around the cells. This is very, very important, because when the cells become turgid, this hoop actually ensure that the cells mostly increase in length rather than the diameter. And also the ends of the two gut cells, they are joined together. Now try to imagine, yeah, guys. Sorry. Now we have gut cell. So this gut cell, they join at two ends. Okay. Now what we want to do here is we want to make the gut cell turgid in the process. So when it turgid, they expand, crown up. Okay. So what we want them to expand, we will want them to expand in terms of diameter of the cells, but expand in terms of the length of the cells. So try to imagine, because these two sides, they are joined, and they cannot expand it in terms of diameter, they expand in terms of the length. So in terms of the length, when they expand, because these two parts, they are joined, so they make it more curved. Okay? Are you clear? So when they expand, they will become more curved. Why? Because the uneven thickness of the cell wall. So the cell wall away from the pore are thin. The one that near to the pore actually is thicker. So the rate of expansions will be different. Can you see that? So it means that when the water molecule enter into the cells through the osmosis down the water potential gradients, what will happen here is the thin part wall expand more compared to the thicker cell wall. So therefore, it tends to curve when the water molecule. And how to make sure that they increase in terms of the length rather than the diameter, they do have this cellulose microfiber actually form something like a hook. Okay, like a hook. Can you see that? Surrounding the cell. So means that in terms of expansions, they have no choice to expand in terms of the length rather than this diameter, because this hoop basically restrict. You try to imagine you tie a string onto a balloon. So when the balloon want to expand, so it can't expand anymore. Why? Because you have the string, okay, around them. So how do they expand? They expand in terms of the length. But when they expand in terms of the length, they do have this. Can you see that? The joint and also the uneven thickness. So make sure that when the cells become turgid, they tend to more curve. Therefore, they open the stomata. Okay, so this is how the structure actually helps in terms of the functions. So you can see that we have a pair of the gut cells, and you realize that the gut cell now also contain chloroplast. Okay, you can write make a notations here, so you can see that the presence of the chloroplast. So it means that the plant cells here can carry out, okay, the gut cells can carry out photosynthesis, but limited to cyclic photophosphorylation only. Sorry. So limited to this cyclic photophosphorylation. Are you clear? So means that the role of the chloroplast here actually is to produce ATP, but reduce NADP, no. Because they only have the cyclic photophosphorylations. Because later, when you look at how the gut cell actually open the stomata or regular stomata, we need to have ATP. So that's why this chloroplast only can produce ATP. So means that they don't carry out the Calvin cycle. Okay, always remember for this chloroplast, no Calvin cycle, because there is no reduced NADP. Are you guys clear about this? Okay, so it means that that's why you look at the gut cell. They are sensitive to the light, because when they receive the light, they can carry out cyclic photophosphorylations and they produce ATP. And because of production of ATP, then you have a series of reactions until the opening of the stomata take place. Okay. So when the gut cell gains the water molecule, always remember, when they gain the water molecule by osmosis, they become turgid, they curve more or they bend more, okay, because the thin wall, okay, the thin wall actually bend more compared to thick wall, then they make the gut cell actually become more curved and open the pore between the two cells. Can you see that? This is the closing. When they expand, they become, okay, they open the pore. Now, always remember, even though we say the closure, it doesn't mean that it closed totally. Yeah, it doesn't mean that it closed totally. So in terms of regulation of the stomatal activity, we can see that they show daily rhythms of opening and closing. So in this case, you can see that if it supplies, okay, for the thick line here represent the darkness and daytime. So the nighttime, daytime, nighttime, daytime, we give a. So we can see that during the daytime, because the plants carry out photosynthesis, so the stomata open. At night, close, open, close, open, close. Can you see that? So it showed daily rhythmics, opening and the closings of the stomata. But if you look at this, if we prolong exposure of the sunlight or light, then you can see that they don't open permanently. Can you see that? They don't open permanently, but they regulate it. Can you see that? Up and down, up and down. But overall, can you see that there is a decrease in the pore size? Are you clear? Why? Because we do have other factors that come into the considerations here. Because first, when you keep on opening, the stomata, what will happen? The plant may lose the excess water as water vapor. Therefore, we need to control. Okay, the plant need to control the diameter. So overall, you can see that because of prolonged, can you see that? Prolonged exposure to the light, then you can see that there is a generally decrease in the stomatal diameter. And this is very important because prolonged opening may cause the water contents, okay, of plants under high rates of transpiration. In this case, in this table, they didn't show us how about the temperature, how about the wind speed. Are you clear? So if you consider all those factors, then you realize that the plants may undergo what we call the water stress. It can't afford to lose too much of water in during a transpiration as a water vapor. Therefore, they have to keep on open, close to regulate. Okay. And prolonged darkness, in this case, also you can see that occasionally open and close. This one also have to remember, even though the plants may not carry out photosynthesis at this moment, but plants do need to carry out respiration. So it means that they need to open so that they get extra O2. Okay. So in terms of this, we need to know that openings during the day times to maintain the inward diffusion of carbon dioxide and also the outward diffusions of oxygen. So this one related to the photosynthesis. Okay, and also our diffusion of water vapor in transpiration. The closure of stomata at night when photosynthesis cannot occur. And also we want to reduce the rate of transpiration and conserve water when the stomata close. So in these diagrams or table, we can see that a summary, what actually caused the stomata to open and close, what kind of stimuli. So if you look at this, increase the light intensity because want to carry out photosynthesis. Therefore, you can see that stomata respond by opening. So low carbon dioxide also because want to carry out higher rate of photosynthesis. So therefore they open. So dark times, no photosynthesis. So no need CO2, right? So in this case, they close. Okay. And then this is something special. High carbon dioxide concentration in the airspace in the leaf. Then you can see that actually stomata respond by closing. Until now, we are not so sure why stomata actually close, but some scientists actually predicted or hypothesized that because of high carbon dioxide concentration, they will cause the presence of these bicarbonate ions. And this bicarbonate ion actually may combine with the water, okay, the productions, okay, of these bicarbonate ions may produce the H plus ion, and the present H plus ion may cause the closure of the stomata. Are you clear? You still remember the carbonic anhydrase? So something similar to this reaction. But until now, scientists cannot actually pinpoint out what is the main mechanism to cause the closure of the plant's stomata due to the high carbon dioxide concentration. But we do know that high carbon dioxide concentration may cause or causes the stomata closure. So low humidity, high temperature, this one because of the transpiration. So if the plant does not want to have too high the rate of transpiration, then they respond to the low humidity and higher temperature by closing the stomata. And as well as water stress in this case, definitely we don't want to have a high rate of transpiration. Therefore, in this case, we say that hormones in this case involved, we call ABA hormones. So these ABA hormones will cause the closure of the stomata. Okay, so the most of their part we will be able to actually analyze them, okay, by relate it to the photosynthesis or transpiration. So in order to carry out photosynthesis, then therefore they have to open. If there's no photosynthesis, definitely stomatal response will be closing. Okay, a closure of the stomata. For transpiration, as we know that if the transpiration rate is too high, then definitely the plant want to close the stomata so that they won't lose too much of the water vapor. Okay. So what is the mechanism of the stomata opening and closing? So in this case, very important, particularly to the light. Okay, what will happen here is because with the light energy, this light energy will be absorbed by the pigments, the chloroplasts here. So therefore, cyclic photophosphorylations take place. So with the cyclic photophosphorylation, we can produce ATP. Now, what will actually happen here is with the release of the ATP or the formation of ATP, now you can see that ATP actually phosphorylates the proton pump. Okay, so first process here, ATP going to phosphorylate the proton pump. So when the proton is pumped out, try to imagine a proton pump out. So outside of the cells now, we call it something like the hyperpolarization in this case. Why? Because outside have the high concentrations of the H+. So try to imagine now, this is outside of the cells, this is inside of the cells. So outside to have higher concentrations of the H+. So it means that outside now become positively charged compared to the inside negatively charged. But the question here is, okay, for the protons to move back is impossible because we don't have the protons, what we call this, the ion channel. So proton cannot move back. But the thing here is outside too positive, inside very negative, they generate this what we call the electrochemical gradient. Are you clear? Electrochemical gradients. So therefore, no choice. Now we need to have a movement of the ions. So what kind of ion can move in? So it depends on what kinds of the ion channel we have. So here we have the K plus ion channel. So it means that now K plus can move in. Are you clear? From the outside of the cells into the gut cell. So the movement of the K plus enter into the cells now will trigger a series of reactions, results in the movements of water molecule now. So we have the aquaporins. So this aquaporin now allows the water molecule to move into the gut cell. Why? Because when the K plus enter into the cells down the electrochemical gradients by facilitated diffusions, what will happen here is they trigger a series of reactions, result in the entry of the many kinds of ions into the cells. So for example, chloride ion enter into the cells, right? Okay, K plus move in, and then chloride ion also move in. So therefore, we have the high ions concentration inside the cells, lower down the water potential, therefore water molecule enter through the aquaporins by osmosis down the water potential gradients. Okay. So you can try the question now. So first, we can see that ATP powered the proton pump in the cell surface membrane actively transport H plus or proton out of the gut cells. So therefore, the lower down the concentration of H plus inside the cells and become negatively charged inside the cells. So in this case, we call it hyperpolarizations. It become more and more negative in this case. So these hyperpolarizations cause the K plus ion channel to open. When this K plus ion channel opens, then you can see that K plus now diffuse into the cells down the electrochemical gradients. So highlights here is not the concentration gradients, but electrochemical gradients. Why? Because outside too many positive ion, inside too little positive ion. So it creates this, what we call the electrochemical gradients. And outside too many positive ions, so positive ion repel each other. And inside lack of positive ion, more negative ions. So this will create the what we call the potential to drag the positive ion. But against only K plus ion channel open, right? So no other ions can move in, only K plus, okay, or potassium. Okay, so what will happen here is when K plus diffuse in, or write down is this by facilitated diffusion. Okay, down the electrochemical gradients. Then what will happen here next? So because of the K plus, so the high concentration of K plus inside the gut cell causes the lowering of the water potential. Now be careful. In actual case, okay, in our syllabus, you just need to mention the high concentration of the K plus inside the gut cell actually lower down the water potential. Okay, but in actual case, the presence of the K plus actually will trigger a series of reaction result. The chloride ions also enter, yeah? Chloride ion also enter. So when the chloride ion enter, they follow that is K plus, then you can see the accumulations of the ions. So therefore, accumulation of ion actually lower down the water potential. Therefore, water molecule move in by osmosis down a water potential gradient. Now, this is very important because the recent mock exam, I realized that some of you still use the words concentration gradient. It's not true for water. We use the water potential gradients. Okay, so when the water molecule enter, there is an accumulation of the water molecules. Therefore, we do know that the entry of water molecule or accumulation of water molecule increases the volume of the gut cells. Therefore, the gut cell now expand. So because of the feature of the gut cells, right? Because they join at two ends. That's one. Second, the cell wall that near to the stoma, okay, or the stomata thicker, and then the one that away from the stomata actually thin out. So therefore, different rate of expansions results in the cells now become when it comes turgid, when the cell becomes turgid, different rate of expansion make this gut cell now tends to curve. Okay, so the thin outer wall expand most, the cell now curve apart, leading to the opening of the stomata. Okay, so how the closure actually take place? So the opposite process actually take place now. So now imagine that now the opposite reactions. Light energy, no. So any absorptions, no. Any cyclic photophosphorylation, no. ATP productions, no. So without ATP productions, now ATP won't phosphorylate the proton pump. Okay, now what will happen here is the inside the cells we always have the H plus or protons to be generated. So it means that when proton concentration increase, okay, so there is no hyperpolarization. Can you see that? There's no hyperpolarizations. And then because H plus accumulated H plus inside, so H plus repel each other, and any H plus ion channel, no proton ion channel, because now this proton pump closed already. Can you see that? Proton pump can't function. When there's no proton pump function, can you see that? H plus start to accumulate. So because of high concentration of the positive ions repel each other, K plus now move out. When K plus move out, it will going to trigger a series of reactions. Chloride ion also move out. Water molecule also move out because now lack of the ions. Correct? K plus move out, chloride ion move out. Okay, so it means the accumulation of the ion now outside of the gut cells therefore osmosis take place, water molecule move out. So it's over opposites. Okay. Now, what we mentioned here is the reductions of the uptake of the CO2 for the photosynthesis. We want to close. Why? Because let's say no sunlight already. Okay, so this is write down, nothing to do with the water stress. Under normal conditions, we still remember rhythmics opening and closing, right? Rhythmics. So it means that at night, for example, we want to reduce the uptake of CO2 for the photosynthesis. So in this case, because we do know that ATP concentration now is low or no no ATP. So hydrogen ion pump or proton pump stop. Potassium ion now leave the gut cells, entering the neighbor cells. So therefore, water potential inside the gut cell is higher because we lose the positive ions. Okay, so therefore, water molecule leave the gut cell by osmosis down the water potential gradients. Therefore, the gut cell become flaccid and close the stomata. So reverse process. Okay, but in the case of water stress, means that the rate of the transpiration is too high. Okay, the rate of transpiration too high, then the plant cells now secretes a hormones called ABA. Okay, it secretes. Okay, so the secretion of ABA will have a significant effects onto the stomata and cause the stomata to close. And because of the closure of stomata, then it reduced the uptake of the CO2 for the photosynthesis. Definitely, photosynthesis will drop. Are you clear? Unless it's C4 plant, different story. Okay, C4 plant, because they can concentrate the CO2 into these bundle shift cells. Okay, and definitely because of the closure of stomata, we can reduce the transpiration. Okay, so therefore we can reduce the loss of the water vapor and conserve the water. Are you clear? Because if you continue, then the plant will lose more water vapor compared to what they can gain from the soil. Okay, so how this ABA actually cause the stomata closure? Now look at this. So what will actually happen here is the ABA now bind to the bind to the receptor. When you bind to the receptor, so it's a cell signaling pathway. Can you imagine that? So when ABA, the signal here, the chemical signaling molecule, bind to the receptor, it will trigger two reactions here. So the first reactions, okay, the first reaction, which is not shown in this diagram. So ABA bind to the receptor will trigger or inactivates the proton pump. Okay, proton pump is inactivated. So if the proton pump inactivated already, then we do know that K plus now move out, water molecule move out. That's the first mechanism. Are you clear? But it's not enough. It's not strong enough in this case. Are you clear? Okay, not strong enough. So second pathway here, ABA, when they bind to the receptor, you're going to cause the openings of the calcium ion channel. So calcium ion enter. So the entering of calcium ion, so calcium ion act as a secondary messenger, will trigger a series of reactions, allowing the chloride ion to move out. Because the chloride ion move out already, you can see the outside now have more calcium, the chloride ion. Can you see that? More chloride ion, negatively charged ion. So you're going to pull, okay, or drag the K plus out. So when K plus out, chloride ion move out, so outside actually accumulates with the ions. Are you clear? So dense water molecule move in. And these calcium ions, as I said, that they also have the reactions, okay, or the effects onto the potassium ion channel. So in plants, actually, potassium ion channel, they have two kinds of potassium ion channel. One allows the outwards diffusion, another one allows inward diffusion. So the one, the inward diffusion also will be blocked or inhibited by these calcium ions. So I summarize the process in these diagrams. Okay, look at this diagram. Okay, so inside the cells, outside the cells. So first thing, ABA. ABA, can you see that ABA bind to the ABA receptor? So with this complex, ABA receptor complex, a hormone receptor complex, it triggers two reactions here. One, inhibits the proton pump. So when inhibit the proton pumps, therefore no proton is or can be pumped out. Second, they also stimulates the openings of the calcium ion channel, so allow the calcium ions to move from outside of the cell into the gut cell. So inside the gut cell, calcium ion act as second messenger. So what this second messenger can do? Second messenger actually can trigger the opening of the chloride ions, allow the chloride ions to move from the cells to the surrounding. And we do know that chloride ions, they are negatively charged. So with the presence of the chloride ions, it makes the negatively charged, what we call this, outside the cells become negatively charged. Inside the cell, because of you stop the proton pump, so we have H+. We have the potassium ion. But can proton move out? No, we don't have the proton ion channel for proton, but because accumulations of the positively charged ion outside accumulation of negative charged ions, then because of these electrochemical gradients, potassium ion now move out. So this is called outward diffusion. And this calcium ions also can trigger a series of reactions to close the ion channel that allow the inward diffusions of the potassium ion, because we don't want potassium ion to move in. Are you clear? We don't want the potassium ion to move in. Just try to imagine outside a lot of potassium ion, then potassium ion move in. What is the purpose? Move in, move out, move out, move in, move out, move in. This is important. So therefore, this calcium ion act as secondary messenger to block, to inhibit this potassium ion channel. But not all potassium ion channel, the potassium ion channel that allows the inward diffusion of the K+. So therefore, you can see that outside accumulates negative charge ions, chloride ions, and also the positive charge ion, potassium ions. So the concentration of ion increase, lower down the water potential. Therefore, water molecule now move from the gut cell into the surrounding or neighboring cells through the aquaporins. Therefore, it causes the cell to be flaccid, stomatal closure take place. Are you clear? Okay or not? Clear. So you can try the question now, activity 18.21. Before I review the answer, it can take 5 minutes. Okay, so look at this hormones called abscisic acid, ABA, has been found in a wide variety of the plant, including the ferns and the mosses, as well as the flowering plants. ABA can be found in every part of the plant and is synthesized in almost all cells that possess chloroplast or amyloplast. Amyloplast is something like chloroplast, but they don't, they don't have chlorophyll, they don't carry out photosynthesis, but what they do, they actually can store. It's a storage organelles. They're storage organelles. They store the starch grains. Okay, they store starch grains. So you can see that plant hormones slightly different from the animal hormones, mammal hormones, because in mammal, when the hormones, we always have a special organs that synthesize it. For example, insulin, glucagon, pancreas. Can you see that? When you talk about this ADH, okay, so we have the one that produce, we have the in the hypothalamus, correct or not? Produced in the hypothalamus and then secretes through the posterior pituitary glands. Can you see that? Okay, so in gains, we talk about these adrenalines, secreted by adrenal glands. So we do have a special organs or tissues to produce the hormones, but not in plants. In plants, you can see that they can have many types of cells that produce the same kinds of hormones. They don't have a specialized organs or specialized tissues to produce the hormones. And one of the role of this ABA is to coordinate the responses to the stress, and this stress is termed as a water stress. So it's known as a stress hormone. So if a plant is subjected to a difficult environment conditions, let's say very high temperature or much reduced in the water supplies, then it responds by secreting ABA. So high concentration ABA stimulates the stomata to close, reducing the loss of the water vapor from the leaf. So therefore, they can reduce the rates of transpiration. Okay. So how exactly ABA achieved the closure of the stomata is not really completely understood, okay? But it seems that gut cells have the ABA receptor on their cell surface membrane, and it's possible when the ABA binds to this receptor, it can inhibit the proton pump to stop the hydrogen ion being pumped out. Okay, so first thing, inhibits the proton pump. So ABA also stimulates the movement of the calcium ions into the cytoplasm through the cell surface membranes, okay, because they have this to open the calcium ion channel. Okay, so by facilitated diffusions. So, and we don't know that calcium ion act as second messenger to activate the calcium, the channel protein to open, allow negatively charged ion to leave the gut cell. So this negatively charged ion is the chloride ion, okay, to move out of cells. And because of the chloride ion accumulate outside, right? So outside become negatively charged, inside become positively charged. So therefore, it will stimulate openings of the channel protein, which allow the movement of the potassium ions. Because you have this what we call the gradients, the electrochemical gradients. So therefore, it allows this potassium ion to move out. So this one right now is the outward diffusion. So outward diffusions of K plus is allowed. Okay, but at the same time, calcium ions also stimulate, because second messenger, closure of the channel protein that allowed potassium ion to enter. So means that the inward diffusion of K plus now is stopped. Are you clear? So means that the plants, they actually have two kinds of the ion channel. They can allow the the ions to move in or move out. Okay, it's more specific in this case. Okay, so the loss of the ion actually increases the water potential inside the cells and water molecule, guys. Write down water molecules. So this cell actually refer to the gut cell, not surrounding cell, the gut cell. Okay, so water molecules now move out to the cell by osmosis down the water potential gradient. So therefore, gut cell become flaccid and stomata now close. Okay. So any more question regarding to this the closure of the stomata?